Hey everybody, my name is Mark Latham, and this video is all about DNA, about how you can find it in the dust on your floor, on your toothbrush, on your dirty dishes, and all those places you can extract DNA, have it unravel the same way that we're seeing here in this animation. This is what the CSI people do, is they use basic principles of microbiology to split open cells, pull out the DNA, and once it's a strand, just read it all the way through. That's what we're covering in this video today.
Now, the thing that a lot of people don't know is that the techniques developed to extract DNA from cells from any crime scene actually have been around for quite some time. Back in the 1860s is when DNA was first discovered inside of a cell. They were trying to isolate proteins because they thought that was the secret for all information being passed throughout life. It was a good guess, but they completely missed out on DNA being the secret. It wasn't until the 1950s that they really put together the digital nature of DNA. But those techniques that have been around for over a century, you can easily do in your kitchen with just house implements. And I'm really excited to show you how easy it is. We're actually going to be extracting DNA from raspberries. We're going to take 8 raspberries and put them in a standard Ziploc bag, our scientific equipment. 1, 7, 8. And then we're going to add all of our different ingredients to help break these cells down. So we're going to add salt, about 2 teaspoons worth, into the bag. And what that does is it introduces ions, and ions float around until they find a charge that they can satisfy. And that helps break things apart, or at least let them let go of each other. And then we're going to take a little bit of water. This is just distilled water. You can use bottled water yourself. But the thing that is most important in this process is actually the detergent. The dishwasher soap, whatever's in your kitchen, works just fine. This stuff is what actually breaks up the cell membrane. The cell membrane, and you just need a decent squirt of it, is made out of 2 layers of lipids, and the detergent interferes with those connections of those lipids, and they just unravel, they fall apart. Also, a lot of the organelles inside the cell are made up out of similar structures, and the detergent breaks those up as well. And the fun part starts. We're gonna mash it up. As you start doing the pressing first, go ahead and just let as much of the air out as you can without making a mess. And then just mush away. You wanna keep mushing for about 5 minutes or so. If you're doing strawberries, which is the age-old example, raspberries, a lot easier to break up. The actual mechanical motion here is doing a lot of good to break up the cells, but not nearly as much as the detergent and salts going in. Hopefully I don't get flagged on YouTube for how graphic this is. What we need to do is we need to take all the big chunks of cell and raspberry out of the solution. And we can do that using coffee filters. So the coffee filter grabs onto everything that isn't below a certain size. I'm gonna pour all this into the 2 filters is what I'm using, stacked. All right, and already it's seeping through the coffee filters. So as it starts to drip out of the filter, go ahead and just have it drip into a clean cup. And if you're impatient, you can squeeze a little bit, but don't squeeze too hard because then you'll rip the filter. All right, once you have enough to fill the bottom of the cup, go ahead and set your slurry solution to the side. Next up is the extraction. We have all this DNA in here, and the backbone of all the DNA molecules are negatively charged, and so they're attracted to water, which helps it dissolve the DNA into the solution. Next up, we're gonna replace those water bonds with rubbing alcohol. We need to introduce this onto the top of the solution, but you can't just mix it up. You gotta do this carefully, which is just simply pouring it down the side. So we'll go ahead and gently pour the alcohol solution, just about as much alcohol as there is raspberry juice. All right. So as you can hopefully tell, the raspberry juice is staying on the bottom, the alcohol is sitting on the top of it, and we'll get some close-up shots here of the DNA forming on the very top. These bubbles are trapped in this sort of snot mucus, which is the DNA precipitating up and out of the raspberry juice. All that snot-like looking stuff right there is actually DNA. Okay, now if we take our solution here and we mop up all the different strands inside the alcohol. And all of this stuff sitting right here on the toothbrush is concentrated DNA with some alcohol still sitting inside of it. Now, it may not be 100% pure DNA. If you were to sequence it and figure out who this belonged to, and you figure out a raspberry in this case, it would actually be a lot of noise. There'd be a lot of information in there that's actually all the other guts of the cell. This is exactly what all the labs do in order to extract DNA from whether it's a hair follicle or whether it's a spit. All the labs use this technique to extract DNA from the substances, and then they run it through specialized equipment to figure out exactly what the A's, T's, C's, and G's say about what the DNA belongs to. You can have your own genome sequenced by tons of different services out there. I personally went with Nebula Genomics, and they've done an outstanding job. If we take a look here, we have an incredible glimpse into the DNA that makes me me, for the better and also for the worse. Nebula Genomics does a great job of putting together a library of curated scientific papers. And they rate each and every one of these aspects that they found in your DNA with all the other people that used their service. So for example, here, if we take up my highest scoring correlations between my genome and a scientific paper, I'm in the top 100th percentile of all users of Nebula Genomics, most likely to be bitten by a mosquito. Also, you can see that my genome has a predisposition to height, and indeed I'm 6'5". But where this really becomes useful for me is the fact that I have a correlation between specific groups of base pairs in my genome that could result in me having something called the Brugada syndrome. If I wanted to look more into whatever this Brugada syndrome, I can view the full report and see exactly where on my genome the correlation ties to. I had no idea that this is something passed down through my family. It is now within my responsibility to go and find a doctor, a professional cardiologist, and have them take a look at my heart to see if this is something that I need to worry about. So it's very easy to become completely overwhelmed with all of these bad traits. Oh no, I'm gonna die of cancer early. The truth is, none of that is written in stone. This just simply gives everybody a leg up for taking care of their own health. I've always known that knowledge is power, and I cannot wait to take advantage of the deficiencies in my genome, as well as leverage the strengths that I've found. We can also see that there's been a correlation found between shaky hands and a specific sequence of alleles in your genome. I've always worried that it was my nutrition, if it was my stress levels that caused my hands to shake, and now I know it's just simply who I am. Now, if we go to my traits associated with my genome and I look at this row, I can see indeed I am nearsighted. I've been wearing glasses for the last few years. But the one thing I really enjoy about having this done is I have a better appreciation for physical conditioning. I've never really felt like I needed to work out that much, but I have noticed that whenever I put in that effort, I get returns very quickly. Within the week, I can see my endurance improving. Just simply knowing that this is something in my genome that I can rely on has been such a great encouragement for me to get back into the gym and actually use this. Just simply understanding that I have this advantage when it comes to working out has done more to get me in the gym than anything before. All right, so hopefully you have an appreciation for the technology that we have today. Previously, it was $3 billion worth to figure out how to get one person's genome sequenced. Right after that, it was millions of dollars, and then over the last few years, the prices dropped all the way down to just simply $300 to have every single base pair in your body sequenced. So even though extracting DNA from raspberries may not directly impact your life, hopefully you have an appreciation for how much this can impact your life through taking responsibility for your genomic health. I fully see a time where all health insurance companies will give you extreme discounts on your premiums if you take responsibility for the information in your DNA. Because if you're being proactive and taking care of these potential issues long before they do become issues, the health insurance companies are going to be paying far less down the road. Now, the cheapest way to keep this type of information flowing to you is to hit the like and the subscribe button. This is going to be the focus of my channel, bringing science to you that impacts your life as much as possible. The more information you have, the more likely you are to interact with the world in a meaningful way. Whether or not that makes your life more rich, that's entirely up to you. Thank you so much for watching my video, and I will absolutely see you in the next one.